Bienvenidos y bienvenidas nuevamente al auditorio virtual de Perú Conciencia, la Feria Científica del Perú en su 9ª edición y 2ª edición virtual. Terminando esta jornada del día jueves, continuamos con la serie de videos que llegan a nosotros gracias al convenio con la Escuela Internacional de Nanociencias Nanoandes. Así que les dejamos aquí este interesante video que nos explicará sobre algunas aplicaciones de la nanotecnología.
Bueno, una vez que hemos sintetizado las nanopartículas de oro recubiertas con sílice mesoporosa, aquí tenemos el resultado. Y este es un polvo que resulta de la calcinación de las nanopartículas para remover el template y quedarnos con los poros formados. Con estas nanopartículas ahora vamos a hacer un proceso, un experimento, para poder medir la capacidad de carga que tienen estas nanopartículas. Y para eso vamos a utilizar rodamina. Hemos preparado aquí dos soluciones. La solución de rodamina, que se encuentra en una concentración de 2 miligramos por mililitro, y la solución de nanopartículas, que también está a 2 miligramos por mililitro. Esta solución de nanopartículas la hemos preparado pesando aproximadamente 10 miligramos de nanopartículas en 5 mililitros de agua. Y lo hemos sometido a un baño ultrasonido para que las nanopartículas se suspendan bien en la solución. Para hacer este experimento vamos a colocar 1 mililitro de rodamina y 1 mililitro de nanopartículas. Y lo vamos a hacer por triplicado para poder tener un mayor poder estadístico. Una vez que hemos mezclado las nanopartículas con la rodamina, vamos a incubarlas durante toda la noche sin agitación y a temperatura ambiente. Bueno, entonces ha transcurrido una noche desde que pusimos a incubar las nanopartículas con la rodamina, y ahora lo que vamos a hacer es pasar a estos tubos de Eppendorf 1 mililitro de la solución y vamos a hacer los lavados. Para esto vamos a centrifugar los tubos. Vamos a recoger el sobrenadante y vamos a hacer varios lavados hasta ver que ya no haya restos de rodamina en el sobrenadante. Ya tenemos aquí los tubos que tienen la solución de nanopartículas con rodamina, y lo que vamos a hacer es centrifugar para recoger las nanopartículas y lavar la rodamina. Para eso vamos a centrifugar durante 5 minutos a 17.500 RPM y lo vamos a hacer varias veces hasta ver que haya una disminución de color en el sobrenadante, lo cual indica que los lavados están bien hechos. Una vez que se han centrifugado los tubos, vamos a poder observar el pellet de nanopartículas y en el sobrenadante la rodamina que no ha sido absorbida. Entonces, este sobrenadante lo vamos a colocar en balones de evaporación de 25 mililitros para después poder cuantificar mediante UV visible la rodamina que no está absorbida y mediante la diferencia de rodamina inicial y la rodamina en el sobrenadante vamos a calcular la cantidad de rodamina que ha sido absorbida. Una vez que hemos recogido el sobrenadante del primer lavado en los balones de aforación, vamos a volver a hacer un segundo lavado y para esto vamos a suspender las partículas en agua destilada. Voy a colocar 1 mililitro de agua destilada en cada uno de los tubos. Voy a darles un pequeño vórtex para resuspender las nanopartículas y voy a volver a lavar, voy a volver a centrifugar. Ahora vamos a medir mediante UV visible la concentración de la solución inicial de rodamina. La solución teóricamente es de 2 miligramos por mililitro. Ahora vamos a comprobar que es así. Para esto vamos a colocar 3 mililitros de agua destilada en las cubetas de cuarzo que tiene un paso de longitud de la luz de 1 centímetro. Y vamos a colocar 6 microlitros de la solución inicial de rodamina. Esta es la solución de rodamina que vamos a emplear. Tenemos 10 microlitros de solución de rodamina en 3 mililitros de agua. Ahora, para proceder a la cuantificación, vamos a usar este espectrofotómetro. Y lo primero que vamos a hacer es medir la línea base, medir la absorción del blanco, que en este caso es solamente agua. Vamos a medir un espectro entre 400 a 600 nanómetros. Y en este punto estamos midiendo la línea base. Una vez que ya tenemos la línea base con la absorción del blanco, ahora vamos a medir la solución inicial de rodamina. En este punto, la absorbancia máxima a 554 nanómetros es de 0.7421. Hasta este punto hemos hecho 7 lavados de las nanopartículas que tienen rodamina en solución. Y bueno, después de cada lavado hemos visto una disminución considerable del color del sobrenadante, lo cual indica que ya la rodamina que resta en la solución es la rodamina absorbida en las nanopartículas. Aquí tenemos los 3 balones de aforación en donde hemos recogido el sobrenadante después de cada lavado. Y lo que vamos a hacer ahora es aforar la solución hasta 25 mililitros para proceder a la cuantificación de la rodamina en el sobrenadante. Ahora, para cuantificar la cantidad de rodamina en el sobrenadante, hemos hecho una dilución en las cubetas de cuarzo que contiene 2.500 microlitros de agua destilada más 500 microlitros de la solución de rodamina del sobrenadante. Con esta solución vamos a proceder a medir y a cuantificar la rodamina mediante espectrofotometría. La absorción máxima es a 554 nanómetros y corresponde a 0.9803.
Bueno, en este punto, para recapitular un poco, lo que hemos hecho es sintetizar las nanopartículas de sílice mesoporosa con un corazón de oro mediante una metodología de un solo paso. Luego, estas nanopartículas las hemos calcinado para remover el template de setup y después hemos utilizado estas partículas para hacer un test para calcular la capacidad de carga de rodamina. Utilizamos rodamina para simular que es un fármaco o es la droga de interés que queremos retener en los poros de las nanopartículas. Para hacer este test, incubamos una solución de rodamina con una solución de nanopartículas, la dejamos toda la noche y el día siguiente realizamos lavados de las nanopartículas mediante centrifugación. Colectamos el sobrenadante y mediante espectrofotometría medimos la absorbancia de rodamina. Ahora lo que vamos a hacer es calcular la concentración de la rodamina en el sobrenadante, restarla de la concentración de la solución madre y con esto calcular la capacidad de carga de rodamina. Entonces, el primer paso es conocer la cantidad de rodamina en la solución madre. El promedio de la absorbancia medida fue de 0.742 unidades de absorción. Ahora ustedes conocen esta ley, que es la ley de Lambert-Beer, mediante la que podemos calcular la concentración de una solución conociendo su absorbancia. Y tenemos que la absorbancia es igual a la concentración del absorbente multiplicada por la longitud atravesada por la luz en el medio, que en nuestro caso fue de 1 centímetro, y multiplicada por el coeficiente de absorción. Reemplazando los términos, tenemos que la concentración va a ser igual a 0.742 unidades de absorción dividido por 1 centímetro y multiplicado por 208 mililitros por miligramos a la menos 1 por centímetros a la menos 1, que es el coeficiente de absorción correspondiente a la rodamina. Ahora, ustedes recordarán que para medir esta absorción de la rodamina, a la solución madre le aplicamos una dilución. Y es importante conocer el factor de dilución que aplicamos. Recuerdan que en las cubetas, en una solución de 3000 microlitros o 3 mililitros, colocamos 6 microlitros de la solución madre de rodamina. Esto nos da un factor de dilución de 500. Y este factor de dilución tiene que ser multiplicado por la concentración calculada. Con esto tenemos que la solución madre es de 1.78 miligramos por mililitros de rodamina. Ahora vamos a calcular la concentración de rodamina en el sobrenadante. Ustedes recuerdan que hicimos este ensayo lo hicimos por triplicado y la absorbancia promedio fue de 0.985 unidades de absorción. Nuevamente aplicamos la ley de Lambertville y tenemos que 0.985 dividido para 1 centímetro por el coeficiente de absorción de la rodamina. Y con esto tenemos que aplicar nuevamente el factor de dilución. Ahora, en este caso es importante aclarar que aquí tenemos dos factores de dilución. ¿Por qué? El primer factor de dilución corresponde a la dilución del sobrenadante en el matraz de 25 mililitros. Ustedes recordarán que en el experimento utilizamos 0.5 mililitros de rodamina y 0.5 mililitros de nanopartículas. Entonces, en una solución final de 25 mililitros, que fue a lo que aforamos en el matraz, le colocamos 0.5 mililitros de rodamina inicial. Esto nos da un factor final de 50-50. Luego, para medir la absorción de la rodamina mediante espectrofotometría, en la cubeta de medición colocamos una solución de 3 mililitros y a esta solución de 3 mililitros agregamos 0.5 mililitros de la solución del sobrenadante. Esto nos da igual a 6. Y con esto tenemos un factor de dilución final de 300. Este factor tiene que ser multiplicado por la concentración y con esto tenemos que en el sobrenadante hay 1.42 miligramos por mililitro de rodamina. Ahora, para calcular la capacidad de carga de rodamina que tienen nuestras nanopartículas, aplicamos estas fórmulas. Primero, para conocer la cantidad de rodamina B cargada, a la concentración de la solución madre tenemos que restarle la concentración de la rodamina en el sobrenadante. Con esta rodamina B calculada, calculamos la capacidad de carga y para eso tenemos que utilizar la cantidad de rodamina B cargada, dividirla por la cantidad de rodamina B cargada, más la masa de las nanopartículas que empleamos, que en este caso fue de 1 miligramo, y todo esto multiplicar por 100. Con esto tenemos 1.78 miligramos de rodamina menos 1.42 miligramos es igual a 0.36 miligramos retenidos en los poros de las nanopartículas. Calculando la capacidad de carga tenemos 0.36 dividido para 0.36 más 1 miligramos de nanopartículas multiplicado por 100 y esto nos da un total de 26%. Entonces, en conclusión, las nanopartículas que hemos sintetizado tienen una capacidad de carga de 26% de rodamina. Eso es todo, muchas gracias.
Vamos a ver por MED las nanopartículas que hemos producido y por eso estoy buscando dónde están las nanos, aquí. Se ven ya nanopartículas que son o sea aisladas así o que hacen aglomerados como esto. Vamos a trabajar sobre estas. Y aquí trabajo con una tensión de 3 kilovolts. Entonces puedo tomar unas primeras imágenes así, que voy a guardar. A cambiar de voltaje, porque si pasamos a un voltaje más alto, podemos atravesar un poquito la muestra. Y cuando se cambia el voltaje también se tiene que cambiar la distancia entre el detector y la muestra. Entonces aquí estoy más bien a 1 centímetro. Y ahora tengo que ir a buscar la muestra. Puntos blancos en el medio de las nanopartículas. Estos puntos blancos son las nanopartículas de oro que están dentro de la partícula de sílice. Vamos a hacer los arreglos sobre unas partículas aquí, individuales, y en particular aquí voy a intentar alinear las partículas. Así tenemos una primera imagen, pero se cargaron mucho las partículas durante los arreglos, entonces se ve muy bien que están blancas cuando las demás están gris. Entonces ahora podemos tomar esta imagen. Aquí se ve que hay 2 o 3 partículas de oro por cada partícula aquí, y si se mide el diámetro de las partículas, bueno, 190 de este lado, y aquí 170. Hay más partículas dispersas. Cuando las partículas están calcinadas, muchas veces es difícil tener una buena dispersión de las partículas y se quedan como aglomerados. Aquí se ve que hay partículas que están un poquito fuera de las partículas de sílice. Con esta imagen se puede hacer estadística porque hay muchísimas partículas. Entonces, estadística sobre el número de partículas que tienen solo un corazón de oro, estadística sobre el tamaño de cada nanopartícula de sílice.
Hi to everyone. My name is Johan Rupioz. I am a research director for the French National Research Center, and I'm working at the CIM lab based in France. So it is my pleasure to introduce this Nanoandes practical, which will be dedicated to the synthesis of a biosensor made of cold nanoparticles for the detection of adenosine. What is a biosensor? A biosensor is a device which is made of two parts. One part is usually functionalized with what we call a bioprobe, which is a molecule that will have some affinity toward a target that will be present in a sample. Indeed, if this target is present in the sample, then it will be captured by the bioprobe located on the surface of the device, and then the binding between the bioprobe and the target will induce somehow the production, actually the transduction of a signal that will be used for the detection of the target. Here are some details about all the different kinds of biosensors you may find around you or in the literature. Regarding the bioprobe, it can be a molecule which might be very different. It might be made of short DNA strands, it might be made of RNA, it might be made of PNA, which is a mixture between peptides and nucleic acids. It might be made of antibodies, enzymes, and so on. Regarding the transduction system, different strategies might be used for the production of the signal. It might be an optical transduction, which is the case in SPR-based biosensors, for instance, or fluorescence-based biosensors. It might be based on a shift due to the increase of mass on the device. This is the case for QCM, Quartz Microbalance System, which is based on piezoelectric device. It might be an electrochemical approach for the transduction of the signal. This is the case for glucometers, for instance. Maybe you know this device, which are used every day by people having diabetes. What's the difference between a microarray and a biosensor? In a biosensor, usually people are interested in the detection of one molecule or one target. In a microarray, actually you can run the detection of a large number of targets using a single device. So this is somehow a parallelized biosensor. Here are the main issues about biosensors and microarrays. When designing these kind of devices, you will always have to focus on the high specificity, high sensitivity, and ability to work in raw or complex media. What is a high specificity? The specificity will be the ability of your device to detect the right target, even though the sample contains other molecules or other targets whose structure and shape is very close to the one you are interested in. The sensitivity will be the minimum concentration you will be able to detect in the sample. So as you can guess, you will always try to design systems where the sensitivity is much lower than this concentration of the target you may find in your sample. The last point is the ability to work in raw or complex media. Indeed, most biosensors are made for working with biological samples, and usually these biological samples are very complex. It might be a blood drop, it might be some saliva, it might be urine, and so on. These samples may contain a large number of different proteins, it may contain cells or cellular debris, and so on. And you have to make sure that the system is still working on these kind of solutions, even though there are many different ingredients in the sample. Here is one of the most common biosensor you may find in a shop. This is a pregnancy test. In the pregnancy test, you will detect a human hormone named human chorionic gonadotropin. So with this system here, you will drop a small volume of urine on the left-hand side here, and thanks to capillary forces, the sample will move this way and will pass over two lines made of antibodies. These guys here are very specific to the human chorionic gonadotropin hormone. And these guys here, which are labeled on particles, are also specific to this target. So if the target is present, everything will be moved this way, and then you will form this kind of sandwiches here, where the target is bound on the surface of the biosensor and also on the label. Okay, so in this kind of approach, it's an optical transduction. You would just have to look at the device to see if there is a positive response or not. And you will have something that will be very specific to the human chorionic gonadotropin thanks to the selectivity of these antibodies. Okay. The sensitivity of this kind of assay is usually about 17 milli-international units per milliliter. This unit here is a unit commonly used in pharmacology. Antibodies are very common and often used for biosensors because they are highly specific to epitopes located on the surface of an antigen. So as you can see here, one single antigen may display different kind of epitopes, meaning you can raise antibodies that will recognize different locations on the same antigen. I guess you all know that these guys have usually this shape. This is the case for immunoglobulins G, with two domains here, which are identical, and that will bind to the same epitope, and one fragment here, which is named constant fragment, that will be specific to a species, that will produce this kind of antibody. There are different ways for producing monoclonal or polyclonal antibodies. Antibodies are very common because they might be raised against different kind of targets, although it's very difficult to have antibodies specific to toxins or antibodies specific to small molecules or drugs. Antibody storage and stability is also an issue, and cost is very important for the production of antibodies. You can see here, this is a very basic and simple scheme describing the production of monoclonal antibodies. First, you have to immunize a mice or rabbit or horse with the antigen contained here in the syringe. Then the animal here will produce specific cells that are displaying antibodies specific to the antigen on the surface. Then these cells will be fused with tumor cells, and then these cells here, named hybridomas, will be selected, isolated, and cultured for the production of antibodies. This is a very long and expensive process. For this reason, since the 1990s, some people are developing other strategies based on nucleic acids rather than on proteins for the production of ligands that might be very specific to a target. These guys are named aptamers. So the objective is to mimic antibodies, but to take benefits of the nucleic acids, which are much easier to produce and easily chemically modified by introducing labels or fluorophores or grafting on magnetic beads, for instance. The production way to get these aptamers is named Selex. Here, in the red circle here, you have the very first example of a DNA aptamer, which is specific to the thrombin protein, which is very important human protein. You can also raise aptamers against very small molecules, which is not possible with antibodies, as I said earlier. Here is the adenosine. So adenosine is a neurotransmitter. It's also a small natural compound which is involved in blood pressure control, and it's also a metabolic intermediate. Here you have the very simple structure of the first aptamer, which has been identified for the binding and specific binding of adenosine. So this sequence here has been published in 1995 and has been turned out to be very sensitive for the detection of adenosine. The KD has been measured and is in the range of 6 micromolar. Here are the crystal structures of the aptamer binding two adenosines. Actually, it's forming a pocket, as shown here on this scheme, and in this pocket, two adenosine molecules can fit and be bound to the wall structure. So here you have the shape of the folded aptamer with the main pocket here, where two adenosine molecules are bound. So today, I'm very pleased to show you this practical for the Nanoandes network. So the objective of this practical will be the synthesis of gold nanoparticles, the coverage of these gold nanoparticles with oligonucleotides, which will be used as split aptamers, and then the use of these nanoparticles for biosensing the solution of adenosine, which has been set as a target in a biosensing assay. So first, we'll do the gold nanoparticle synthesis. You will see it's a quite simple step that will require only 3 reagents: water, gold salt, and sodium citrate. But before I get started, I just want to tell you that you have to wear lab coats, gloves, safety goggles, and work into a safety chemical fume hood. So the setup is quite simple. You have here a heating system. In the balloon here, there is 50 milliliters of water, of boiling water. And on the top of this flask, there is a cooling system. This cooling system is cooled by flowing fresh water into it. So the water comes from this pipe here, up through this system, and goes here into the sink. You have to make sure that here you properly tighten the flask here, but at this position here, it has to be loose to make sure that it can freely move in case of problem. Okay? So before we get started with the practical, I will show you how to use an automatic pipette and also how to prepare some reagents, like for instance, the sodium citrate solution. Okay, so now I will explain to you how to use an automatic pipette. Okay, the systems are made of 2 parts. This one is an automatic pipette, and then you have to use disposable plastic cones. These ones are single-use systems. So you plug the plastic cone this way on the automatic pipette, and then you can adjust the volume you will need by turning the button here in this direction. Okay, if you turn this way, you will decrease the volume you can pipette, and if you turn the bottom the other way, you will increase the volume. You can see here through this window the volume which has been set. So here, let's say I need 50 microliters. Of course, I use an automatic pipette which is in the range of the volume I need. For instance, on this one, I can pipette up to 200 microliters. So usually, the systems are made to pipette volumes in the range of 1/10 of the value which is written here, up to the value which is written here. The plastic cones which is used here is made for aqueous solutions. It means that if you want to pipette organic solvents, like ethanol or acetone, you may have leaks because this plastic is not made and suitable for the organic solvents. For pipetting, it's very easy. You just have to press this button. Actually, there are 2 levels. This one is the first level, which is the one which corresponds to the volume which has been set here. And then if you press further, you will have a second level which will be used to release the total volume of the liquids which is contained into the pipette cone. So for pipetting, you just have to first press till the first level outside of the liquid, then move the pipette cone just few milliliters below the level of the liquid, then gently release the button and fully release it, and then take the flask where you want to transfer the volume, move the pipette cone close to the wall of this flask, and then gently release till the first level, and before leaving, press fully the button till the second level. And then you're done. So now we'll prepare the citrate solution. So the citrate solution is made of this powder, which is sodium citrate, 3-basic dehydrate salt. Okay, so we'll weight some of this powder to prepare a 100 milliliter solution at 1%. 1% meaning 1 gram of this powder per 100 of milliliter of water. So of course it's way too large. I mean, we'll not need 100 milliliter of powder. We'll only weight 50 milligrams that we will dissolve in 5 milliliter of water. Then it will make a 1% solution of citrates. $65 milligrams. Then I will adjust the volume to make sure that I will have the 1% solution, meaning that I have to dissolve these 65 milligrams into 6.5 milliliter of water. So everything is ready. I have my safety equipment. I can start the practical. So first, I will use this automatic pipette and pipette 50 microliters of gold solution. This solution is quite stable. It can be prepared in advance. So you can see that it has a bright yellow color, which is very characteristic to any gold salt solution. Then I remove the lid and add the gold solution into the flask. After that, I can use the citrate solution, which has been prepared just before the experiment. It is very important to make sure that you do not use an old solution and that you use a solution that has been only freshly prepared. So for that, I will quickly add 1.9 milliliter of citrate solution dissolved in water at 1%. 1% meaning 1 gram per 100 of milliliter of water. Now it's done. So you keep the heating system on, okay? And then you can observe the color shifts that will happen within the first few minutes after addition of the citrate. You can also start to record the time, making sure that the experiment lasts at least 30 minutes up to 1 hour. So the color is changing a little bit now. It's getting bluish or purple. And then it should turn as a deep red solution. and eventually it's turning red, which is a typical color of any gold colloidal solutions. So now it has been 30 minutes since we launched the reaction. So the reaction is completed. We can cool down the solution before using it. So to do that, you first switch off the heating system, you lower the wall system, and then don't forget to switch off the water flow through the refrigerating system. Well, so now we will record the visible absorbance of the colloidal solution. So as you have seen, it has a deep red color. So this is a colloidal solution which has been cooled down at almost room temperature. So first, I have to launch and define the conditions for the recording of the absorbance. So I use absorption spectrum, and then I have to set the minimum wavelength. So let's say 300 nanometers. And the pitch, let's say 2 nanometers. Maximum wavelength, 650. And that's it. And then I can launch the acquisition. I have a message that says that I have to first prepare the blank. So as the reaction has been carried in an aqueous solution, the blank can be recorded with deionized water. So this is what I have here. I have the cuvette here. So this one is compatible for small volumes. For instance, 500 microliters are sufficient for recording the spectrum. Then I can press OK, and it will scan the whole spectrum and use the references at the blank. So now the blank spectrum has been recorded. I can prepare the solution for the colloidal solution. So this is the gold nanoparticle solution. I can pipette 500 microliters once again. And then I can launch the acquisition for the sample. So here, let's say these are gold nanoparticles. These gold nanoparticles are quite safe. There's no specific things to take care of, okay? Just make sure that you have the proper bin for recycling or disposal of the solutions. So the maximum wavelength is about 520 nanometers, which is consistent with gold nanoparticles in the range of 20 nanometers in diameter. Now it has been completed. So the absorbance at the maximum wavelength is about 0.98 absorbance units. So now the synthesis of the gold nanoparticles is completed. The gold nanoparticles are covered by citrate ligands, which is present on the surface of the nanoparticles. But before the coupling with the aptamer fragments, we have to displace the citrate molecules and to add phosphine, which will be way more convenient for the conjugation of the gold nanoparticles with the oligonucleotides, which contains the silyl moiety. Okay? So to do that, we have to add the phosphine, which is this reagent here, and we have to adjust the amount of phosphine depending on the concentration of the nanoparticle. Well, so we have to adjust the amount of phosphine to displace the citrate. Okay, so you remember that the absorbance of the gold nanoparticles at 220 nanometer was 0.98, okay? So it's about 1, okay? So this value here is very important. So it means that 1 milliliter of gold nanoparticle solution has 1 optical density units at 595 nanometers. So for each 1 unit here, we have to add 3 milligrams of phosphine, which is also named BSPP. Okay? We have 50 milliliters of this 1 optical density unit solution, meaning that we have to multiply 50 by 3 milligrams and then weight 150 milligrams of phosphine. Okay? So here is the phosphine. In this piece of paper, there are 150 milligrams of phosphine. And then I will add the reagents. Into the flask. Add the lid. And then I will maintain the stirring for at least 2 days at room temperature.
again, everyone. So it has been 2 days since we launched the displacement of the citrate with the phosphine. So here is the colloidal solution containing the gold nanoparticles. And now we'll have to remove the citrate excess and the phosphine excess to resuspend the whole sample in a simpler solution containing just water and the phosphine. So I will split the sample into a series of Eppendorf tubes. I will split about 2 milliliters in each of them, and then I will centrifuge the samples to remove, after that, the supernatants. So well, now samples are ready for centrifugation. One very important thing, before using a centrifuge, make sure that you properly balance all the samples so that there's no problem during the centrifugation process. So everything is ready. All samples are properly balanced through the sample holder into the centrifuge. I can put the lid and launch the centrifugation step. So it would last 10 minutes at 15,000 Gs. Well, after centrifugation, all the samples are gathered together into one single tube, okay, to give this very concentrated solution containing the gold nanoparticles covered with the phosphine. Then we have to record the absorbance of this solution, and more specifically, the absorbance at 520 nanometers, which is usually the maximum wavelength for the absorbance of gold nanoparticles in the range of 20-25 nanometers large. Well, so as you have seen, we used a 50 times diluted solution, and we recorded the absorbance at 520 nanometers. The absorbance is 0.85. Because we have 50 as a dilution factor, we can calculate the expected concentration in terms of optical density of the stock solution by multiplying 0.85 by 50. So it means that the expected concentration would be in the range of 42.5. Now we will do the oligonucleotide coupling on the nanoparticles, and we will use oligonucleotides that contain a thiol moiety that will react very easily with the gold layer. In a sample, in an Eppendorf tube, we will first add 50 microliters of thiolated oligonucleotide, which has been diluted to 10 micromolar. Okay, so we take 50 microliters of this solution. Then we add a small amount, meaning 50 microliters of BSPP2, which is a more concentrated solution containing the phosphine. Okay, this solution is made of 20 milligrams of BSPP diluted in 400 microliters of water. Then we'll add the solution containing the gold nanoparticles. The objective is to have a potential optical density in the range of 10 after dilution in the sample. The total volume for the reaction is 500 microliters. So if I want to calculate the initial volume of my stock solution here, I have to multiply the final concentration with the final volume, meaning 10 optical density multiplied by 500 microliters, and then divide this product here by the initial concentration, meaning 42.5 optical density. The result is 118 microliters of the stock solution of gold nanoparticles. And then, because the final volume is 500 microliters, I have to measure to calculate the amount of PBS, phosphate buffer saline solution, to add to this mixture of reagents. To do that, I have to subtract the volume of each of the components to the final volume. 500 minus 50 microliters of oligonucleotides plus 50 microliters of BSPP2 plus 118 microliters of gold nanoparticles gives 282 microliters of PBS. I will mix all these reagents and leave them to react on the bench for 1 hour 30 up to 2 hours at room temperature. And because at the end I will have several samples made of adenine 4, which is one of the oligonucleotide, mixed with cuts, which is another oligonucleotide, with another sample made of adenine 8 oligonucleotide coupled to cuts, which is the last oligonucleotide. You understand that I will have to prepare several samples here containing either the adenine 4, either the adenine 8, or either the cut oligonucleotide. And because the cut sequence appears in both reagents, I will prepare one sample of adenine 4, one sample of adenine 8, and 2 samples of oligonucleotide cuts. Well, now we'll prepare the samples. So first, I will add the oligonucleotide. So 50 microliters. I take adenine 4 sample. I put it in an empty vial. Then I will add 50 microliters of the BSPP2 solution, which is here. Then I will add 118 microliters of the stock solution of the gold nanoparticles. And then I will add the PBS, 282 microliters. Here we are. So I prepared the sample for adenine 4. Okay, so it's slightly bluish. Okay, color should change later, no worries. And then I will repeat the operation for adenine 8 and 2 samples of cut Aptamer. So here are the 4 samples I prepared. Okay, I will leave them on the bench for 1 hour and 30 minutes up to 2 hours. Okay, no stirring, no heating. So the reaction is done now. The oligonucleotide have been coupled on the surface of the gold nanoparticles. And just to make sure that all the gold surface has been reacted and will not react anymore, we'll saturate this gold layer with a solution containing PEG thiol. So PEG PEG means polyethylene glycol. This is a long polymer which is highly hydrophilic and that will help in the saturation of the gold nanoparticle surface. So to do that, I prepared a solution containing the PEG solution dissolved in water at 150 micromolar, and I will take 35 microliters of this solution that I will add in each of the 4 samples I prepared earlier. "35 microliters. In each of the 4 samples. So now the 4 samples are ready. We will leave them on the bench for 1 hour up to 1 hour 30 without any stirring at room temperature. And after this incubation, we will remove the excess of thiolated oligonucleotide and thiolated PEG by centrifugation. To do that, we'll do it the same way as we did before to remove the excess of citrates by using a centrifugation step for 10 minutes at 15,000 Gs. So the reaction with the PEG thiol is done now. So each sample can be moved into the centrifuge. Don't forget to properly balance all your samples. Well, now the reaction is completed. So we centrifuged the samples. We will remove the supernatant and then resuspend the pellets in a Pipettes solution. So you can see the deep purple or red pellets at the bottom. I remove the maximum volume of supernatants. And then suspend the sample in Pipettes. And now I get the deep red color. Okay? You will repeat this step twice to make sure that you totally removed the excess of oligonucleotide. And then you can store the sample at 4 degrees before running the biosensing assay. So during the last step, we used the nanoparticles and we grafted 3 different oligonucleotides on them. One sample was made of the split adenine 4 aptamer. The other one was made with the split aptamer adenine 8. And the last one is complementary, and it's made with the cut aptamer strand. Okay? So these guys have been grafted on the nanoparticles. Then we added a PEG thiol entity to make sure that we fully saturated the surface of the gold nanoparticles. And then we centrifuged all samples several times to make sure that we were removing all the unreacted reagents. And now samples are ready. We can run the assay. So to do that, we'll prepare a series of samples. Some samples will contain the adenosine at different concentrations. One sample will contain the uridine. So uridine is a small molecule. It looks like adenosine, but it's not. Okay, so it has a similar molecular weight. The structure is very close, but it will not fit into the pockets that will be formed by both split aptamers. Okay, this is very important. It will ensure that we can test the selectivity of the assay. Okay, and the last control will be made with the adenine 8 aptamer, which is much longer. It's named 8 because there are 8 base pairs that can be formed between the adenine 8 aptamer and the cut aptamer. These 8 base pairs, they are forming a stem made of a double helix DNA, and it will be very stable. Okay, it will be so stable that it will hybridize even though there's no target. So it will be used as a positive control. Let's do the preparation of each sample. So here is the first sample. This one contains the adenine 4 aptamer and the cut aptamer. There's no adenosine. You can see it is pink, regular color for a colloidal suspension of gold nanoparticles. This one contains the same oligonucleotides grafted on the particles, but it also contains 0.5 millimolar of adenosine. The third one contains the same oligonucleotides with an increasing concentration of adenosine at 1 millimolar. The fourth one contains 2 millimolar of adenosine. And the fifth one contains 5 millimolar of adenosine. Okay? So this is the range of adenosine we will test. But besides this sample, we also have 2 more samples. This one is made of the same oligonucleotides, adenine 4 and the cut aptamer, okay? And it also contains the uridine, 5 millimolar of uridine. And the last one contains different aptamers. It contains the adenine 8 aptamer with the cut aptamer, okay? So all these guys will be put into the centrifuge, and we will launch the centrifugation for 10 minutes at 15,000 Gs. So the centrifugation step is done. We recover the samples. As you can see, in each of them, there is a dark pellet, which is made of the nanoparticles which have been centrifuged. For sample 1, if I shake this pellet, then I recover the pink solution corresponding to the colloidal suspension of gold nanoparticles. Okay, it makes sense because there's no adenosine, so there's no aggregation of the nanoparticles. For the second sample, it's pink as well, a bit lighter. Okay. For the third one, it's still lighter. Okay, so remain a small pellet. For the fourth one, there's no resuspension. Okay. Or almost no. For 5, there is a pellet, okay? You get a colorless solution, meaning that there is a full aggregation of the nanoparticles. Sample 6 is made with the uridine, okay, which is not recognized by the split aptamers, and that should not enable the aggregation. And indeed, we recover the pink color, which is characteristic to the full colloidal solution. For the control, it's fully hybridized, so we get the pellets, okay, and almost no color in the colloidal solution. So you can see that even though we do not use any UV visible spectrophotometer, we can have an overview of the assay and get an idea about the concentration where we got the aggregation depending on the concentration of adenosine. If I take the highest concentration of an adenosine, the one with 5 millimolar, so you can see that I have dark pellets at the bottom, okay? If I take this sample, and if I use hot tap water, I will increase the temperature of the sample, and I will destabilize and dehybridize the structure, and I should recover the pink color, which is characteristic to the colloidal solution of nanoparticles. I can do the same with the sample which contains the adenosine 8 aptamer with the cut aptamer. So a few seconds, let's say like 10 seconds, under hot water. And then you can see I can recover the red color, which is characteristic to the concentration, to the solution containing the colloidal solution of nanoparticles. So this process is reversible, okay?
Bueno, este video llegó gracias a la Escuela Internacional de Nanociencias Nanoandes, que participa en Perú Conciencia. El día de hoy les agradecemos por haber estado aquí acompañándonos a nosotros en Perú Conciencia, y los esperamos mañana nuevamente a partir de las 10 de la mañana en el auditorio de Perú Conciencia, que tendremos la charla Tecnologías para nuestra identidad digital, que nos contará Renier. Pero recuerden que la feria está abierta a 24 horas, así que los invitamos a que visiten todos los proyectos de investigación que tenemos tanto en la zona de Perú Conciencia como en la zona de Eureka. En el auditorio nos despedimos por hoy y los esperamos mañana a partir de las 10.